Hi everyone, my name is Ismar Biberovich from Tetra Tech, and I want to start this video by thanking you all for participating in monitoring Benthic Cyanobacteria in the Lake Champlain Basin. We really appreciate your time, your effort, and experience in the field. This video will consist of two parts. This first part, I will be here in my office going through the sampling kit that you have received or should be receiving in the next few days. And then the second part of the video will be a video of me sampling using this particular kit to show you how to use each of the components in the kit. So let's start by talking about the first item in the kit, which is the nitrile gloves. We are all familiar with these. This is protective equipment. Please always wear your gloves when you're handling any type of biomass that you think is benthic or any kind of cyanobacteria, because some of them can produce toxins and irritate your skin. The most important part of this kit are the 4 bottles that you'll be receiving. This blue bottle here, a 250 milliliter bottle, will be used to analyze total toxins in your samples. This is the only bottle that has to be frozen as close to the collection time as possible. You will also be receiving this tiny red bottle. We will be using this bottle for microscopy samples to essentially have some sort of record of each sample we collect. This will be used just to confirm Benthic cyanobacteria in our samples. And then you will be receiving these larger 1 liter white bottles, green and yellow. These are used really for the same purpose, which is to filter 2 liters of sample, or as much as we can, for DNA analyses. And once we filter, then the filters will be sent to a lab we are working with, so they can tell us what cyanobacteria are found in the samples. The next thing we have on our list are these long tweezers. These are simply used to retrieve samples or grab any substrate you may not be able to reach with your hand. They come pretty handy. They would be used maybe to transfer biomass into your bottles if you can't grab them with your gloves if they're too slippery. We also have these scraping tools just in case you find substrate and detaching cyanobacteria or biomass from that particular subject. Let's say a rock is not that easy. You can use these to and scrape them along the surface of the substrate to detach them, and then maybe use the tweezers or your gloves to collect the biomass. We have also provided this grabber tool. This one collapses, as you noticed in your bags, and this is simply used if the substrate is too deep and you want to collect it. This adds about 3 feet of length, so you may be able to retrieve it. So you will be receiving this as well. We've also provided this rope. Each of you should have received a 6-foot piece of rope. This is just for a reference when we designate those 6x6 quadrats that you will be sampling in. Again, these will be fictional quadrats. We didn't provide you with an actual 6x6 quadrat with some maybe tubes, but that's just sort of the general area in which you will be sampling and answering most of the questions from the Benthic form on. So again, in the next part of the video, I will guide you through how to use this equipment when sampling in the field. As always, if you have any questions, you are welcome to reach out to me or Lindsay about those. Feel free to call us or email us. See you in the next part of the video. Alright, we are at the Sandbar State Park, and the first step of the process after you arrive to your site is to do the broad visual scan of the shoreline and the shallow nearshore zone. Not only is this where Benthic cyanobacteria are most likely to accumulate, but also where people are most likely to interact with water. As you watch me here, I'm carefully looking at the sand, logs, and aquatic plants for any signs of mats. And as you know, these can appear dark green, brown, black, or even reddish sometimes, and may be attached to submerged surfaces or floating near the shore. I'm also checking for any unusual slimy, maybe, or leathery textures on the shore, and even dried patches above the waterline. The visual assessment is critical. Even if no mats are found, documenting the conditions helps us understand where and when benthic cyanobacteria are likely to appear. After this step, please be sure to fill out the online visual assessment form to record your observations and take photographs. After you have finished scanning your site, the next step is to fill out the online visual assessment form. You'll do this every time you visit your site, whether or not you find benthic cyanobacteria. Some questions are required each time, and others only show up if you think mats might be present. You can also upload photos to help confirm what you saw, or ask us if you're unsure whether it's benthic cyanobacteria. At the very end of the form, there's space for any extra notes, observations, or questions. Feel free to use it to share anything unusual or anything that didn't quite fit into the earlier questions. You can access the form using the QR code shown here in the video or from the printed guide in your volunteer packet. These submissions help us understand when and where mats are showing up and what visual environmental factors might be contributing to their appearance. Feel free to pause the video now and visit the survey to get familiar with it. It is finally time to collect samples, but before we do that, it's important to review the volunteer guide to determine which sampling scenario applies to your situation. In this case, I have waders on and I'm pretending to have identified a floating benthic cyanobacteria mat that's accessible by wading, so I'll be following scenario 4. The guide itself outlines step-by-step instructions for each scenario, including safety reminders, equipment use, and how to handle samples. Reviewing the guide ensures consistency across all volunteers and helps us collect high-quality data. Now that I've confirmed the scenario I'm going to follow, I'm putting on the nitrile gloves provided in the kit. These protect both you and the sample from contamination. Always be cautious when entering the water. Watch for slippery rocks and unstable footing. If the site seems unsafe, please do not sample it, just document what you see and that is going to be sufficient. In this next part, I'm just using the provided 6-foot rope to get a rough idea of what 6 feet looks like and to imagine a quadrat in front of me that I'm going to be sampling and answering the questions based off of. Using the grabber tool, I gently stir the mat to simulate natural disturbance like a swimmer or dog moving through the area. This helps release material into the surrounding water. I'll wait about 10 or 20 seconds to let it mix. Next, I take the blue bottle and submerge it just below the surface near the disturbed area. I fill it to about 20%, close it, shake it, and discard the water. Then I refill it to about 90%. This sample will be used for toxin analysis and must be frozen as soon as possible. Then I take the green bottle and fill it near the densest part of the mat. If there are multiple areas with similar density, feel free to subsample from each. This sample, along with the yellow bottle collected next, will be used for DNA analysis. Both will be filtered through the same filter and analyzed together, so there's no functional difference between them. Both the green and yellow bottles should be refrigerated after collection. If the mat is cohesive, I use tongs to gently lift some of the material and place it into the yellow bottle. If it's too diffuse to grab, I submerge the yellow bottle into the mat until it fills about a quarter of the way, and then I repeat the same process for the red bottle, which will be used for microscopy. Please note that if the mat is cohesive, you can also do this for the green bottle. As I mentioned earlier, functionally there's no difference between green and yellow bottles. To get a representative sample, you can repeat this step from 3 to 5 different spots within the quadrat. The goal is to fill both the yellow and red bottles. Once you've collected your samples, it's important to follow the final steps outlined on pages 14 and 15 of the volunteer guide. First, make sure to complete the online visual assessment form the same day you collect your sample. Next, store your samples properly. The blue bottle must go in the freezer, while the green, yellow, and red bottles should be kept in the refrigerator or a cooler with ice. If you don't have access to a fridge, keep the samples in a shaded, cool location until pickup. To arrange sample pickup, contact either myself at Tetra Tech or Lindsay Carlson at the Lake Champlain Committee. Our contact information is listed in the guide. Thanks again for your participation. Your observations and samples are essential to helping us better understand and respond to Benthic Cyanobacteria in our lakes. You have finally reached the end of the video. We want to thank you for taking the time to go through our training. In the video description below, there is a link that will take you to a Microsoft form. And this is a very brief form that will just confirm that you have reviewed all the training materials and that you are ready to go out into the field and sample. If you have any questions, if any concerns arise, if anything is unclear about the guide, please reach out to me and Lindsay, and we'll do our best to get back to you with an informative and hopefully a helpful answer. See you guys around.